In this video we're going to be looking at the replication of DNA. Now DNA replicates prior to cell division, and it does that using this enzyme that we've got here called DNA polymerase. We're going to go on and look in more detail as to how this process occurs. Now at this point, if you can't remember your structure of DNA, it might be worth just going back and having another look at it. But you should remember that your DNA is composed of two complementary strands. One strand has got the 3' end here and the 5' here, and the other strand has got the 5' here and the 3' here, and that's important to remember, as will become clear as we go further along. So first of all, for replication to take place, the DNA unwinds itself and the hydrogen bonds break between the bases, and that separates this two strands, so you end up with two template strands. So here's our two template strands here. Now I said in the first slide that an enzyme was involved called DNA polymerase, and it is involved in the replication of the DNA. Now the DNA polymerase can't just start the replication process, it needs something called primers. Now a primer is a short strand of nucleotides which binds to the 3' end, and once that primer is bound to there, so this part here we could see is a primer, once it's bound to there, the DNA polymerase is able to add nucleotides to the 3' end of the new DNA strand using complementary base pairing. So what happens is the DNA polymerase starts to add the complementary base pairs. And you'll see the same sort of thing happening on the other strand, although we'll look at that in more detail just shortly. And the primer's added and the nucleotides are added, the complementary nucleotides are added by the DNA polymerase. And you need to remember that hydrogen bonds will form between these complementary base pairs. Once that's occurred, sugar phosphate bonds will form here and here on the new strands that are there, and you end up with two new DNA molecules that have been formed, but they're identical to the parental molecule. Now the title on this slide here is called the leading and the lagging strands, and we're going to look at this in more detail in the next couple of slides. Now it's important to remember that DNA polymerase can only add DNA nucleotides in one direction, and that is from the 3' end of a growing DNA strand. In other words, DNA polymerase using complementary base pairing adds the DNA nucleotides on to the deoxyribose 3' end of the new DNA strand which is forming. So that results in something which is called the leading strand being replicated continuously and a lagging strand being replicated in fragments. Now to explain that, we're going to look at some of the diagrams on the next slides. So let's look at this slide to have a look at, first of all, the replication of the leading strand. Now the leading strand takes place using this DNA strand here as a template. And you'll remember you've got the 3' end here and the 5' end here. Now that means that this new strand that is formed here will have a 5' end down here, and the 3' end will be up at the top. So what happens first of all is the primer attaches itself to the template strand. Then the DNA polymerase can add on DNA nucleotides using complementary base pairing to the 3' end. In other words, this is the 3' end here. So the bases will be added, complementary obviously, and at the 3' end. So you'll see here you've got the start of the complementary strand or the new strand of DNA being produced. And it's being produced by the DNA polymerase adding the nucleotides on at the 3' end of the new strand that's forming. So because we've got this travelling in this direction, in other words from the 5' to the 3', and we can add the nucleotides on at this 3' end, the replication of the leading strand can take place continuously. Now in this slide here we're going to look at the replication of the lagging strand of DNA. Now that takes place over at this side here using this template strand. Now we need to remember that we've got our 5' end here and our 3' end is up here. Let's move this down a little bit. So that means that the new strand that is being produced will have a 3' end here and a 5' end here. Now, remembering that DNA polymerase can only add on nucleotides from the deoxyribose 3' ends, we have a problem because our 3' end is down here. So what happens is a primer will attach itself as this replication fork that you can see here is forming. As these template strands have separated, then what happens is a primer attaches itself, say here, and then that means that the DNA polymerase, can then add the nucleotides in this direction, in other words the 5 to 3 direction, adding them on the 3' end here. So you get a section of DNA that's been made, and then another primer attaches itself further up as the DNA is opening up, and DNA polymerase can add on the nucleotides, and then another primer, and so on. So you get what are called these fragments here that are formed. Now these fragments are then joined together by another enzyme which is called ligase. So you end up having all these little fragments that are produced in this 5-3 direction, and then they are joined together by the ligase enzyme. So let's just have a look at this diagram, which gives us a kind of overall view of the process. So the first thing that happens is that the DNA will unwind itself. The hydrogen bonds will break between the nucleotides, the bases, breaking into the separate strands. This strand here is our leading strand. So what happens is you get the primer attaching itself here, binds to the 3' end of the DNA template. So it binds here, and you get the DNA polymerase adding continuously the nucleotides on, because this is the 3' end here. So you're going in a 5-3 direction here, adding the nucleotides on at the 3' end of the new growing strand, and the leading strand is formed. The nucleotides form hydrogen bonds to the complementary base on the parent strand. So we get the hydrogen bonds forming, and the sugar phosphate backbone forms. On the lagging strand, though, you'll have ended up having a primer, say, added itself here, and then what will happen is DNA nucleotides will have been added on from the 3' end here, and you'll have then another primer and another fragment formed, and then your DNA ligase will join these fragments together, and then you'll have another primer added here, and from the 3' end you'll have nucleotides being added by the DNA polymerase, and so on. And the fragments, you remember, are then joined together by the DNA ligase. There's a couple of names in here that you don't need to know about, primase, and you just need to know the term primer, and you only need to know the term fragment. You don't need to know this word here, Okazaki fragment. It's just a name that's given to them because of who discovered them. And to finish up, in order for DNA to take place, the nucleus must therefore contain DNA to act as a template. It must have primers, it must have a supply of the 4 types of nucleotides, DNA polymerase and ligase enzymes, and it also needs a supply of ATP, that is energy.